Hi, Dr. Biology here. I hope you're all good. This is Epistasis and this is for A-level biology, specifically AQA, but it does cover other exam boards. So without further ado, let's get started. Okay, so you're probably wondering why am I showing you a picture of the Happy Mondays, apart from they are a pretty epic, legendary band of the 80s. They did a song called Step On. Well, they didn't. They did a cover of a song called Step On. And epistasis means step on or stand on. Okay, so as it says here, epistasis arises when the allele of one gene affects or masks the expression of another in the phenotype. This can happen when genes act in sequence by determining the enzymes in a biochemical pathway. So what we say is that a dominant allele, it codes for the production of a functional enzyme. Now, if there is different steps within the production of, for example, pigment or fur colour, there might be several stages to go through, and therefore several genes might be involved. So as it says here, if alleles of either gene are recessive, then the enzyme will be non-functional and the biochemical pathways cannot be completed. The example I'm going to use today is mice fur colour. So as you can see, we have three colours of mice. Albino, which is not actually a colour, it's just albino, so there's no pigments. Then you've got black, where there is a pigment. And then you've got this thing called agouti. Now, agouti is when another enzyme is involved in terms of making the pigment, the black pigment, be black and yellow banded hair. So you can see I've got an example down here. So this is actually dominant to black. Now you'll see there's two genes, C and A. So what has got to happen is that if for an albino to be a black, so to have a black pigment, you need to have a dominant gene or allele, dominant allele in C. And then if that black colour is going to then finally be agouti, then the enzyme has of the second gene allele has to be dominant to get black and yellow banded hair. Now, if the parents have the enzyme allele recessive, then they won't produce the pigment. So in this case, if the offspring had recessive alleles, they would all be albino. And then with black, if they would be black if the enzyme allele was A, because they would be non-functional. Okay, so you'd have non-functional enzymes. Okay, to summarize, so the agouti coat color, the brown color that we were talking about earlier, okay, is dominant. So AA is dominant to solid colored fur, which is recessive AA, which is the black. However, a separate gene, C, is necessary for pigment production. So a mouse with a recessive C allele at this locus is unable to produce pigment and is therefore albino, regardless of the allele present at locus A. So what genotypes produce albino mice? Well, as we've just said, that it has to have recessive C allele. It doesn't matter about the A alleles. So you would get that mix. So even though there's dominant A's, okay, and even recessive A's, if there's a recessive allele C, then they will be albino. A common question is then to say make a cross between heterozygotes for both genes. So in this case you can see heterozygous parents, and then it says state the phenotypic ratio. So the first thing you would do is obviously you need to specify the genotypes. So you need to, it will be a 4x4 in this case. So you can see that it is exactly the same for the male and the female. And then you would do a simple cross, okay, between the two. So you can see the crosses there. And then you need to work out the phenotype. So you can see the phenotypes there. We've actually got 9 agouti. I'm just counting them. Yeah, 9 agouti. We've got 3 black and we have 4 albino. So the phenotypic ratio would be 9 to 3 to 4. Okay, so 9 agouti, 3 solid colour, 4 albino. So in this case, we know that the C gene is epistatic to the A gene. So it's masked by the A gene. Okay, I'm going to go through a couple of exam questions now, so this will help you understand the principle behind epistasis, although I'll be honest, these questions also contain information about sex-linked genes as well. Okay, so let's have a look at this first one. So it says coat colour in Labrador dogs by 2 different genes. So each gene has a dominant and a recessive allele. The 2 genes are inherited independently, but the effects of the alleles interact to produce 3 different coat colours. So that would be your first clue to say this is epistasis, as it's saying that they interact to produce 3 different colours. So the table gives 4 genotypes and the phenotypes, genotypes and the phenotypes they produce. So they give you some information, and then it says what colour coat would you expect each of the following genotypes to give. So we've got a homozygous dominant B, heterozygous E. So if we look on the table, we can actually work out, well, where is there a dominant B and E? So it must be black. Okay, it can't be any of the others because it's got the dominant allele, so it must be black. Next one is recessive B, so recessive Bs, and then we've got a dominant E. So again, looking, it's not yellow, so it must be chocolate. Okay, so next questions asks you, it says that a heterozygous B and E male was crossed with a homozygous recessive female. Complete the genetic diagram to show the ratio of offspring you would expect. Okay, so I'm going to switch now and show you that on a whiteboard. Okay, so a BBE, so BBEE male, okay, was crossed with a recessive homozygous female. So complete the genetic diagram to show the ratio of offspring you would expect. Okay, so we need to think about the gametes first. So looking at the gametes, we're going to be looking at the following. So B, big E, and then B and little e, and then little b, big E. Just going to move that out of the way because that's in the way. There you go. And then for my final gamete, so it's little b, little e. Okay, and then the gametes for this one here are dead easy. It's 4 of these basically, so I'm only going to do it once. Okay, so what I need to do now is cross them. Now I could draw a 4x4 Punnett square, but there's no need because this one here is repeating 4 times. So I'm just going to put that one at the top. Okay, and then just put my gametes down here for the male. And then just cross them, big B, little b, big E, little e, big B, little b, little e, little e, little b, one big E, little e, little b, little b, little e, little e. Okay, so we've got one chocolate, so it has to have both both enzymes, sorry, gene E and gene B need to be dominant to allow them to have the dark pigment and then the chocolate or black coat. And then this one is going to be black and the other 2 are yellow. And the reason they're yellow is because of the first gene for coat color B is actually recessive, homozygous recessive. So that would be your answer. This is the second part of the question, and it tells you the yellow coat colour is due to the presence of the pigment pheomelanin. Can I say that? No, I cannot. Pheomelanin in the hairs. So the black and chocolate coat colours are due to different amounts of another pigment, eumelanin. So that's deposited in the hair. So the more eumelanin there is, the darker the hair. The diagram shows the action of genes E and B in producing the different coat colours. Okay, so you've got the yellow pigment, you've got gene E, which is a functional enzyme, so it's a dominant E, producing more dark pigment, so eumelanin, and then a second gene B, which affects the amount of eumelanin deposited, so if it's dominant then it will be chocolate or black. And it says use this information to explain how the genotype homozygous recessive produces a yellow coat colour. So you've got to work out why it would be a yellow coat colour. Well, the clue is in there that you need a dominant allele to have a functional enzyme. So if it's a recessive allele, then it's going to be a non-functional enzyme. Therefore, no pigment will be deposited in the hairs. So there we go. So no enzyme is coded for when there are no dominant E alleles. Or you could say pheomelanin is not converted, so it remains yellow. The next question then goes on, says the genotype heterozygous dominant B and E produces a black coat colour. So again, you need to talk about the fact that the dark pigment gene E enzyme is allowing the depositing of dark pigment, okay, and then gene B affects how much of it is deposited, and so large amounts are deposited to produce a black coat colour. So E results in enzyme producing eumelanin, and B allele, so more eumelanin is deposited in the hairs. Okay, so this second question is about sex linkage, and then it does go on to epistasis. So a sex-linked gene controls fur colour in cats. Ginger-coloured fur is controlled by the allele big G, and black-coloured fur is controlled by the allele little g, recessive. Some female cats have ginger and black patches of fur that are described as tortoiseshell. Male cats cannot be tortoiseshell. So what is meant by a sex-linked gene? Well, it's a gene that's located on the X or Y or on one of the sex chromosomes. Okay, so a male cat with the genotype X little GY mates with a tortoiseshell female, give the phenotype of the male. Okay, so X little g, so black coloured fur is controlled by the allele G, so therefore it must be black. Give the genotype of the tortoiseshell female. Well, again, so it's X something, so it's going to be heterozygous dominant, so X big G, X little g. Then says complete the genetic diagram to show the genotypes and the ratio of the phenotypes in the offspring of the cross. So we'll go again to the whiteboard. Okay, and finally, so this is related to epistasis. So it says the effect of the big G and the dominant G and recessive G alleles is modified by another gene. So this gene is not sex-linked and it has two alleles. So the allele recessive D changes the ginger colour to cream and the black colour to grey. So the dominant allele D does not modify the effect of dominant G or little g. So it tells you a cream coloured male cat mated with the black female, so they've given you the black female. Male kittens of two different colours were produced. Complete the genetic diagram. So again, I'm going to go over to the whiteboard. Okay, so for this question, they give you the male parental genotype, and the female one they don't, because that was B2. So the genotype of the tortoiseshell female is X big G, X little g. Okay, and then they want the parental gametes. So that's quite simple. So that's XG and Y, because it's a sex-linked gene and XG, XG. And then you quite simply just do a Punnett square. So put the female at the top. There we go. So X big G, X little g, X little g, X little g, X big G Y, and X little g Y. So the question then says, well, that you've worked out the offspring's genotypes, that is them, and then the phenotypes. So you need to specify which is which. So that's the black female, that one. Tortoiseshell female, we already know, so it's that one. You got the ginger male, and finally you got the black male. Thank you, Star-Lord. Okay, so we've got the correct gametes, we've got the offspring genotypes, which is shown in the Punnett square, and the phenotypes, so that will get you the 3 marks. Okay, and finally, so this is related to epistasis. So it says the effect of the big G and the dominant G and recessive G alleles is modified by another gene. So this gene is not sex-linked and it has 2 alleles. So the allele recessive D changes the ginger colour to cream and the black colour to grey. So the dominant allele D does not modify the effect of dominant G or little g. So it tells you a cream coloured male cat mated with the black female, so they've given you the black female. Male kittens of 2 different colours were produced. Complete the genetic diagram. So again, I'm going to go over to the whiteboard. Okay, so we need to work out what the gametes are. So for the male, I'm going to start over here. So X big G D, and there's another X big G D, and then the Y D, Y D, and then for this one is X little g big D. Okay, you could have it twice, but I'm only going to show it once, and little d. So what I can do now is a cross. Now it does want you to work out the male genotypes and phenotypes, but I am going to just draw the whole thing. Okay, so bear with me. Make sure you draw your Punnett squares large enough. Okay, so obviously I didn't need to do those. Okay, because they're the same. So there we go. Right, so let's look at the males, because we want to work out well, that's the genotype. We need to then work out what the phenotype is. So this one here is going to be a black male, okay, and this one here is a grey. There we go, and there are the answers. I hope you found that useful. There's going to be more Inheritance A-level videos coming shortly. Please do subscribe if you haven't already, and I will